We're really excited. Thor Misko with the WSS Foundation Board. Really excited to have with us Dr. Zuberi from JAX Labs, and then also Dr. Tomas Bjornsson, who's working on our mouse model research project from University of Iceland. We also have members of our scientific advisory board and our board of directors that are joining us today to hear and have a kind of informal discussion on just an update of where we are at and where we're going. And before we begin and kind of do some quick introductions where everyone will introduce themselves, just wanted to once again say thank you so much for all the work that you're doing. We're so excited to see the progress that's been made so far, and really excited to see where this is going to continue to go. So thank you from the bottom of our hearts, from the WSS community, for all that you're doing. So with that, I'll turn it over and just one by one, maybe Barbara, just a quick introduction, and then I'll point to the next person on my screen.
Oh, you want me to say who I am?
Correct.
Oh, okay. I'm Barbara Mijon. I'm on the faculty of the Department of Genetic Medicine at Johns Hopkins University. And I'm a geneticist and a pediatrician. And I'm interested in X chromosome inactivation primarily.
Barbara is a member of our WSS Scientific Advisory Board. Andrew?
Quickly unmute. So hi everyone, whoever's watching. My name's Andrew Croxford. I'm also a member of the WSS Scientific Advisory Board and also a WSS parent since about 4 years, soon to be 4 years, I would say, in the end of March. Yeah, so my background is more molecular biology and immunology. And yes, of course, very happy to have this update from Aamir and from JAX and from Hans, and yeah, looking forward to the discussion. So I'll let the others say a few words.
Great, and Chris?
Hello, everybody. My name is Chris Schilling. I'm a member on the Scientific Advisory Board here. I'm a Vice President in Regulatory Affairs and Quality at Forge Biologics. I've been working in drug discovery, particularly in the genetic therapeutic medicine side for about 15 years. Prior to joining Forge, I spent the majority of my time at Nationwide Children's Hospital, where we made some discoveries on genetic therapies and moving programs from early phase to late phase clinical trials, particularly some of the programs like the Zolgensma Novartis program that led to discoveries for spinal muscular atrophy and is now a marketed program, along with other programs that went into Sarepta Therapeutics and other opportunities to generate therapies for children with unmet medical needs. So appreciate time. A close friend to Christina and the community out here, and happy to provide some insights. So thank you for letting me join.
Great. And Christina, since you were mentioned.
Thank you. Good to see you, Chris, and everyone.
Good to see you.
Yeah. So I'm Christina O'Keefe. I am a member of the board of directors for the WSS Foundation, and I'm a liaison to the scientific advisory board alongside Thor. So I helped establish the advisory, I guess, 2 years ago. And so it's so exciting to see where we are today, particularly with this research grant. And I've been on the board since 2017. So to see where we've come in so few years to today is just really exciting. And I follow my passion because my middle son has the syndrome. And so that's why I really am doing the work that I am as part of the foundation. So thank you all for what you do for us.
And Kim?
I'm Kim Lagravenese. I'm also a fellow board member. I'm an attorney by trade, so I tend to do a lot of the legal work that the foundation needs, and any other work that comes up. I'm here to help, and I'm just here to today because I'm excited about listening and getting an update on the research project.
And then our fearless leader, Libby.
Good afternoon, everybody. Dr. Zuberi, it's so nice to put a face to the name. We've heard so much about you over the last year. So thank you, everybody, for being here, because I know with everybody's Zoom schedules, it gets hectic, but appreciate everybody making this a priority. And I just realized when Andrew said how long he had been part of the community, Francesca was my 8-year-old was diagnosed 7 years ago this month. So it feels like a very long time and a short time all wrapped into one. So excited to finally be to this point and hear what the latest updates are here.
Great. And then Dr. Bjornsson.
My name is Hans Bjornsson. I'm a clinical geneticist. I've been affiliated with the foundation kind of because of clinical interest, but I'm thrilled to being able to work on Wiedemann-Steiner from a research standpoint. I think we came up with a very ambitious project, I think, and I was grateful to be connected to Aamir because he really has had some fantastic input into this project. And I've also been able to recruit a fantastic student, Tinna. She'll introduce herself a little bit later, so she's on the call. But I'm interested in this group of disorder. Wiedemann-Steiner, Kabuki, they're pretty similar disorders in many ways. And I'm hoping that some of the principles will be able to translate from one to another. So thank you.
Thank you. And Tinna?
Hi, everybody. Thank you for having me in this meeting. So I just started working for Hans this January, and I was fortunate enough to get this project, which is very ambitious, but I am very excited. I just started my master's in biomedical sciences. So I'm very fortunate to be here today.
Perfect. And then Dr. Zuberi, the floor is yours.
Well, hello, everybody. It's a pleasure to meet everybody. Names I'm aware of, but it's always nice to put faces to it. And Andrew and Hans, of course, it's a pleasure to see you both again. We've had most of our interactions have been fairly regular over the past year or so. Just to introduce myself to everybody, I'm Aamir Zuberi. I'm a lead scientist in the model generation area of the Rare and Orphan Disease Center that we have here at JAC. So it's an NIH and also MMRC funded center. The MMRC is an opportunity that we take advantage of in the many mouse strains which are deposited in the lab fall under a specific financial program known as the MMRC. People buy mice. The lab makes a little bit of excess over the top of the cost of raising the mice, but we funnel that back into the Rare and Orphan Disease Center. So people buying mice for one rare disease ends up funding the next rare disease onward. So it's a way that everybody pays forward, and it's really made life very interesting in the last 6 years or so. We are a full service kind of organization in the sense of, whereas I focus on the model development area, there are other colleagues in the group that focus on the in vivo pharmacology, gene rescue, AAV9 therapies. So what I want to do today is just kind of review the progress that we've made in the development of the models, outline some of the plans that we have in how we want to move forward with the models, and then open it up for discussion. And I think I'm going to need to have the screen sharing enabled for. I just have about 10 slides or so, and I think that's just going to give everybody a quick overview of where we are. I could do it verbally, but you'd get lost in all of the numbers. Thanks, though. I've got the slide here. Let me share. Okay, you should all be seeing the slides now. So just to start off, this is a project that really came to our attention with a call from Andrew. And after a fairly extensive conversation, we really found it was an exciting project and one in which we really wanted to be involved as an institution. And this, of course, is all familiar to you. So I will move straight to the mouse, which is really the speciality of the Jackson Lab. We're not a clinical research center by any means. We focus primarily on development of new mutations in mouse models that accurately reflect the human condition, or as close as we can get, bearing in mind that mouse and human are different species. They have a lot of genes in common. There's a lot of similarity in how these genes function. There are some species-specific differences, and generally we see that in various rare disease models that we've studied. In large part, I think, because the rare disease genes as a class tend to be really important for embryonic development, early childhood development. So just to kind of start with where the literature is on this. So of course, others have published on the fact that loss of KMT2A, which is the causative gene here that we're looking at, leads to embryonic lethality in the mouse. They die around embryonic day 10.5 to 11, which is around mid-gestation. And the COMP consortium has published a number of observations about the various mouse models that they've made. They were a globally funded consortium, primarily though by the US, and Japan and Europe, with a very ambitious goal of trying to mutate every single gene in the mouse genome. They're still trying to reach that goal, but it's always nice when they've identified a mutation in a gene and they actually have an early phase phenotypic evaluation of these mice. So just to kind of let you know what they have found, of course, they found consistent with the previous publications that these mice, when you delete both copies of the gene, the mice are embryonic lethal. And looking over here on the right at the growth curves, the ones here in brown, these are the growth curve of males. The lower brown is the growth curve of females from around about 7 weeks of age. This is probably young adult equivalent in the clinic, through to sort of middle age, around 15 weeks of age or early middle age. And you can see that there's a slight reduction in the body weight of heterozygous males and heterozygous females. The differences here is certainly not statistically significant, but certainly indicates a trend that there is a reduction there. When they actually do drill down to more detailed phenotyping, they see that in heterozygous males, so these are wild type with a null allele, so heterozygous for the null allele, they have decreased total body fat. In both males and females, they see abnormal retina morphology, suggesting that there's vision issues here. Of course, homozygous lethality, and heterozygous females have decreased bone mineral density, and there's increased body mass in both heterozygous males and females, and they're all around the early adult stage. So this is a very quick, very rapid evaluation, but it just kind of represents some key biomarkers that might be of interest as work goes forward on other models that are generated, or even the same model looked at in more rigorous settings. I kind of like to start at the end and then work my way back to the beginning. So this is, if you fall asleep or I go way over your head for the rest of the talk, let me just show you this data here. So these are the brand new mouse models that we made in support, working with the Wiedemann-Steiner Syndrome Foundation. So we've generated a clinical allele, the C1186Y, and I'm using the mouse nomenclature here, which is a conversion of a cysteine to a tyrosine. And we've established that mouse with a unique identifier here, which is 34844. What we find with these mice is that they are small, but they appear active, and they're fertile, of course, as well. And just to kind of overlay the level of analysis that we tend to go into. These animals are made in high throughput production facilities. We have very limited access to these mice. They're handled by colony specialists that work with mutant mice exclusively. Our main concern is that we want to reduce the exposure of any mice that we generate to unwanted pathogens, toxins, or environmental changes. So even something as small as a balance is missing from that room. It represents a potential source of contamination. And so the observations are purely visual. So the mice are small because they look noticeably small. How small is something that we would have to explore at a later date. We've made a model that we call 34611, which is a Fluxed Exon 2. So what this is a wild type allele. There's no mutation, but we flanked a critical exon with 2 LOXP sites. And what would happen is that when this mouse is mated to a mouse strain that expresses CRE, which is an exogenous protein that would then cause a deletion between adjacent LOXP sites. So this can convert a wild type allele into a mutant allele. And this is useful for looking at tissue specific effects as well as temporal regulation of loss of function. And we've also captured in that same experiment just a loss of that exon. So this would be kind of imagining what it would be like in a post-CRE environment, that is that we've deleted the mutation. And what we find here is that the homozygous mice are lethal, which is what we expect, and we can only maintain them by mating heterozygous mice to the wild type. And in working with Hans and his interest in these genes, we've established two new models that are both essentially one model that we've established independently of each other. This is because CRISPR, the technique that we primarily use, is potentially prone to random errors. We sequence all of these alleles that we generate, but of course, when you're sequencing, you only sequence what it is that you're looking at, and there may be other changes elsewhere. So typically what we try to do, and we've done these here as well, we generate pairs of lines, and we look to see how similar they are to each other. If they behave similar, then we have a better confidence that these mutants that we've made are truly the ones that we intended, and they have no additional mutations elsewhere in the genome. So these mice are still in progress. They're in an N1 to N2 expansion. And just to describe this terminology, when we do CRISPR, we generate a founder mouse. This founder mouse has many, many mutations in the same mouse. And then we breed that mouse to the N1 generation. This confirms that we have germline transmission of the mutation, and it also allows us to identify the subset of N1 mice carrying the mutation that we want, and we discard the rest. And therefore, and then when we take these few mice, we will now want to expand them. So we back cross them again back to the inbred strain to generate a larger N2 population. And the mice will be heterozygous for the mutation of both the N1 and N2 generations. This allows us to breed them, and then we can separately evaluate what their homozygous phenotype is once we have sufficient mice to begin those studies. And I'll describe what this model is, and that's why we have a CRE mating also that we're planning on doing with these mice here. So let me now individually go through each one of these mutational alleles, and I think this is where the science is going to get a little bit technical, but it's okay. I've already given you the summary, so that's the only thing you need to remember. What we find with the C1186Y clinical mutations that we've made in KMT2A is that, as I've said, these mice are small and active, and they're viable and fertile. What we find when we do a het-by-het mating is that we're seeing evidence of lethality. Now, this describes the sequence where the mutation is made. So in a normal mouse, the sequence is TGC. This encodes a cysteine codon, and we converted that G into an A. And CRISPR is a great technique that allows us to make these very precise genetic changes in the genome. The numbers are right over here on the left-hand side. So you can see that in the generation of N2 progeny, we're seeing about a 50-50 transmission rate here. So the mutation in the heterozygous state seems to be transmitted as expected. About half of the animals are going to carry the mutation. In our het-by-het mating, normally we get up to 50. This is still in progress, but we're not seeing any homozygous mice at all. Now, this is standard genotyping. These mice are going to be around about 10 days or so after birth. So it's possible that they were born and then lost immediately, or it's possible that they're lost embryonically. Typically, what we see in mice that are born is that the colonists can sometimes get to the dead pups or the carcasses, and they still sample them, and no dead pups were submitted for genotyping, and that leads me to believe it's more likely that this is either extremely early lethality after the mice are born, or that they die in utero during embryogenesis. Moving on to the Fluxed Exon 2. So this is the critical exon. When exon 2 is missing from the gene, the protein essentially is mutated to the point where nothing is expressed at all. And in red here, I've indicated where the 2 small LOXP sequences were inserted, flanking exon 2. And you can see here that we've got about a 50-50 transmission rate in heterozygous state. And looking here, this is a little bit less than the 25% that we'd expect for homozygous, but certainly not unusually low, and it's not statistically significant from 25%. And we're now breeding this strain as homozygous to homozygous, which is what we'd expect, that we have absolutely no defect in these mice, which is critical enough to affect viability. And this is now a good model to use tissue-specific or temporal-specific loss of KMT2A function. And this is the deletion that we acquired during the experiment in which we were generating the fluxed allele. It's very similar to what the effect of the exposure of the conditional knockout allele to CRE would give you as well. So it's a good early indication of what that phenotype is. Interestingly, so these mice have only one allele of KMT2A when in the heterozygous state. And that deletion, that hemizygous deletion exists essentially from birth. When we mate these mice, what we're seeing is a significant reduction in transmission of the mutant allele into heterozygous animals. And this is statistically significant. So, haploinsufficiency, that is perhaps a slight reduction in the total amount of KMT2A expressed, has an impact on the viability and transmission of that mice. And so we're losing essentially about 38% of our hets are lost. So we're getting close to 2/3. So it's not insurmountable. It's just something to be aware of, that there is going to be a very early effect in these null alleles. And I haven't gone back to check the literature if those that were making null alleles saw the same effect. But if they captured the data in the same way that we do, then almost certainly they've noted that also.
Can I ask a question?
Of course.
Just curious if there's been any clinical observations in those animals. Have you been able to note anything from the heads?
No, this is primarily just looking at animal husbandry. So the way in which we approach this is we make the mutation. We're doing extensive sequence characterization to eliminate unwanted alleles, making sure we've got what we intended to get. And then looking at animal husbandry, because our primary concern is how are we going to maintain these mice? How are we going to distribute them? And once we get that and we get some baseline numbers, this is now at a point where we can go to the next stage. And that's something that we've had discussions with in the past. We can utilize grant-funded money and MMRC-funded resources to make the mice. but we can't use the same resources to characterize these mice. That becomes a separate question. Typically, and we can talk about this on my last slide, but we would make these public at this point, and then either you as the research community or new investigators can interrogate that, or we can do it here at JAX, working with the foundation as part of a collaborative research agreement. So again, we're seeing home lethality, loss of function. So that describes the three models. There was one model that we tried to make that we couldn't, and I think it's because of size limitations in the technology. We were thinking about trying to rescue murine KMT2A mutations with the human transgene, and so we took the cDNA, but it's a very large gene, as you can see here. In the mouse, we're seeing about just under 4,000 amino acids, and humans just a little bit larger than the mouse. And we tried to make a single copy insertion into the ROSA 26 locus, which allows us to avoid the issue of multiple copies of a transgene expressing that then by itself can also have a phenotype, which is unwanted. It was just too big in our hands. We tried this about 2 years ago. It might be worth looking at again in terms of capabilities always improving, but at the time we really tried to get a regulated human transgene into the mouse, and just the size of the gene itself was prohibitive. But with Hans's model, he was kind of when he suggested this, I was really excited because it's kind of another way of trying to look at the situation. Our conditional knockout mouse model is wild type until you delete exon 2, and then it becomes a mutant. What happens when you're thinking about a mutant and then you change it so that it becomes a wild type? So this is a similar model where we use LOXP, except that we use it in a slightly different way. So this is a cartoon of what that model looks like. So the KMT2A gene is right here. This is actually intron 1, so it's a non-coding region of the gene. And we dropped into it 2 LOXP sites, so they're here in green. And in between those 2 LOXP sites are multiple repeats of a transcription stop. So what this does is when you drop this into a gene, it really just stops that gene dead right there. And you don't see any expression downstream of that. We've characterized this particular version of the SV40 polyadenylation stop sequence, and it's in triplicate. We've looked at it in other models, and we find it's really rigorous at stopping expression. There's probably a little bit of leakiness, but certainly nothing that we've ever detected at the protein level. And so the model was to insert this lock stop cassette into intron 1 of the mouse gene, which is right here, that would essentially create a null allele. But we've generated that before. The difference is that this null allele can be regulated, so that if you now expose the mouse or the developing embryo to Cre protein, that would cause a deletion between these 2 LOXP sites, removal of this transcription stop, and now the gene is going to be expressed. So it's having a mutant model that becomes wild type at a specified place and time of an investigator's choosing. So it's a great model, and I really appreciate Hans discussing it with us and letting us really try to make this. And I can say that we've been successful. We've generated 2 lines from the original experiment, 2 independent lines. Like I said, we have a daughter 1 and a daughter 2 just because it's a complicated allele, and we want to make sure that they both behave similar. We've confirmed that there's germline transmission, so we're now beyond the founder. We're now having the allele in mice, and this mouse would behave as a het null. And what we have here is that we're seeing fairly good transmission here of what we call our primary line. And we call that our primary line just because we have more of them. And our secondary line, which has slightly reduction in the number of mice that we have, just because the original founder from which it was derived didn't transmit as many alleles as we wanted to. And that's just sort of chance effects there. And we have additional mice in genotyping. So this is the N2 het population is normally what we would start distributing here. And while we're collecting these animals, we're also going to be confirming that in a het by het mating that these mice should really be homozygous lethal. The only other comparison that I can make to these numbers here is what our true het null phenotype looks like. And remember, we saw that transmission distortion. We see a reduction in the number of hets. Our numbers here are really small, so it's not possible to say what our final outcome is going to be. But clearly, we'd be kind of hoping for something similar to what we've seen here using 71 mice as we build our numbers up. That we should see a bias in the number of wild type mice which are generated relative to the number of het mice. But certainly even at this level, if this data is transferable to these strains, we'll have sufficient mice to both distribute to Hans and also to continue some of our characterization to add to Hans's data set, so that by the time Hans does get his mice and establish his colony, we should have a really good understanding of what this mutation is and how it behaves when we mate the mice. And that leads me to my final slide here, which is really the next step. So our goal is to make 34844, which is the strain which carries the point mutation, the clinical allele in KMT2A, flip that strain to public. Right now, they're all private. And therefore, they're not viewable via the Jackson Lab website. And we do this until we get to a point where we establish the biology of the mouse, where we have good confidence in what the genetics of that mouse is and the mutation that it carries. And we also are planning to flip the Fluxed mouse here, the exon 2 allele, to public also. We tend to be a little bit iffy on taking this strain to public. It was generated as a byproduct of this experiment here, and it's easy to generate this model from here, except if the foundation has interest or can anticipate experiments where multiple Flux genes might be in the same mouse, then it might be of value to have this mouse. The post-Cre allele of this mouse will have one LOXP site remaining. This mouse is a deletion with 0 LOXP sites remaining. So there is a technical difference between the two, but we certainly have cryopreserved all three, and we can pull it out as needed. On Hans's strains, they're currently private and will remain so because they were made as part of a funded project until Hans decides that he wants to donate them. He's free to donate them to any resource. If he donates them to the lab, then we can flip them to public here. But of course, there are other European resources as well where mice can be deposited. So at that, I'd like to conclude the presentation and really thank everybody for listening and for your attention. It's been a real fun project. In terms of our potential goals that we could do as well, these mice have really not been characterized with respect to protein expression, with respect to where in embryogenesis the lethality occurs, what is the basis of the transmission distortion. Is it if we look in embryos, do we see 50-50? And then we see a bias towards wild type survival after the mice are born or just before. And also we have the capabilities to take these models and start mating the Fluxed allele with different Cre drivers. They could be brain specific, liver specific, kidney specific, or it could be that we knock out the entire gene just after the mice are born and ask how long do they live and what is their biology. Some of that is probably going to overlap with Hans's work, and so clearly Hans will be the driver here in terms of what his plans are. But I think that there are multiple directions to go, and if the foundation would like us to continue to be involved in up to and including potential options for base editing and gene therapy, we're certainly happy to discuss that. Thank you for your attention.
Oh, wow.
Thank you.
Yeah, thank you so much, Aamir. Fantastic. I mean, really, I hope it's clear to everyone what they've done for us.
Impressive work. That's hard work. Yeah.
when I saw that these mice are viable, I really, that you can actually, the breeding is sustainable. This is such an incredible step forward, because this is a very attractive aspect of this for people who will actually come to work with this strain in the future.
I think so.
That you're able to do this, because this can really be a hurdle for people who take on a project when they realize that they're going to be confronted with very serious difficulties in breeding a strain. Sorry, you're getting a bit of glow there. That, I'm absolutely thrilled to see that the heads are not just active, but also capable of sustaining the lineage. So that's really, really great news.
Yes, there is some notation literature that het females may not be fertile for as long as heterozygous mice carrying mutations usually are. We really haven't seen that evidence yet, but typically at JAX we rotate the het females relatively quickly. They get to about 6 months of age and we rotate them out, but clearly that's an avenue which can be looked at as well. The smaller size of the mice and the fact that the COMP consortium via the IMPC website, the link that I gave you earlier, suggests that heterozygous mice have a phenotype, does allow you to look at the het mice that carry the C1186Y mutation, as well as het null mice, to really explore that in more detail. These mice will be available. There's no restrictions with respect to any investigator can take them, any pharmaceutical industry can take them. The Jackson lab doesn't have reach through in these lines, so there are no restrictions in that respect. Any drug discovery or any use of these animals for the development of therapeutics, the lab also does not have reach through into that. So we try to make these available, as widely available as we can, without restrictions.
That's great. I think also you've got options here. You've achieved a variety of different models. So that's
And so the opportunities for mixing and matching are very favorable as well. You can consider models where you take the C1186Y mutation and have it over the Fluxed allele. That would be a heterozygous mouse, but then you can now inactivate the wild type allele in a tissue or a time of your choosing, and now look to see what the C1186Y mice look like when there is no wild type allele there. Sometimes, and we've seen this in other rare disease models, there's a crisis in embryogenesis. If you can overcome that crisis, then you can end up studying the biology of mice as young adults. And I think one could make an argument that many of the affected children here have survived that crisis, and as young children, they are now reflective of the ability to overcome that. So even though genetically they may score as a particular allelic combination that might be lethal in the mouse, we can now overcome that deficiency by tweaking the mouse just a little bit to get viable progeny being born that we can now use in drug studies.
And just to say, I think we're getting close to shipping. I think the last I heard was mid-March. I don't know if that's changed.
I think that's a separate discussion we can have, Hans. We have these mice and they're available now. I think you ordered a few other mice as well, and you wanted to consolidate shipping. So then the ship date is moved to the slowest of the lines, which I think was a cryo recovery. So if you're willing to split the order, then these mice can be shipped to you sooner.
Yeah, I think we had come to the conclusion just to split it into two.
Okay, I'll have a discussion. That is handled by our portfolio managers, and so I will raise the issue with them, and they can reach out to you and discuss how you want to split your shipment.
Great. Just to, obviously we're a little bit behind schedule, but there have been many. It's been a pretty crazy year. I'm actually, and it's a very ambitious. I think it's an ambitious project. I wasn't even sure that it would work, and so I'm thrilled that we have mice and we have 2 lines. So that's 2 shots, because I think. A very important question that we can answer, and there are many others that Aamir just talked about that we'd love to participate in answering or help with answering. But I think it would be so important to show that there is some effect of postnatal recovery, because I think that would set this disorder apart from many other disorders. You know, the dogma has been that intellectual disability is not treatable postnatally. And to have that, you know, and there's many things that can still go wrong, don't get me wrong, but to have that answer would be valuable, because it would open up anyone with a therapy would then say, hey, should I jump on the Wiedemann-Steiner bandwagon? Look, they already showed this. Maybe I should bring my treatment to that disorder. So, that's what we're going to focus on. But my hope is that Aamir was kind enough to also send us with no cost conditional Fluxed mouse. My hope is that I can use this funding to get more funding and ask other questions too. But with this grant, I think that's the focus that we're going to try to do.
I completely agree with you, Hans. I think our real goal here at the lab at the Rare and Orphan Disease Center has really been to do the heavy lifting in terms of make the models. And now you're essentially at the top of the cliff. And what we'd hope is that established researchers can use the models now to get more funding and hopefully to recruit new investigators into this as well. And I see Tina is already on her way as a new investigator in this area. So growing the field is an important characterization. The other thing is, you know, interactions between different rare diseases. So I think one of the functions, and forgive me, you probably know far more than I do about the biology of this gene, but certainly it has the ability to interact with SET. And we have independently generated SET binding protein mutations, which is, you know, falls under a different rare disease. You know, does a protein which binds SET and other proteins which bind SET, do they have the potential to interact with each other? And can that consolidate rare disease analysis from different foundations, because they're working under the same umbrella, they're looking at the same biology just from different viewpoints. And so I think that there's a synergy there as well. And you know, where we see it, we try to you know, make the foundations aware of it, because sometimes it's not obvious from the foundation name or its biological function. But it's also fairly certain that the biology as described in the literature is often incomplete, and there's new biologies to uncover, and that's what these models can be used for as well.
So if I understand, then some of the next steps here is, you know, obviously getting some of these models in the hands of the researchers to begin doing that characterization and getting some evaluations on how these are performing, characterizing them. Is there any you can share on some of the plans going forward with that?
Certainly. So our immediate plans are to make the strains, the Fluxed allele and the strain carrying the point mutation, public and accessible. Then it can be ordered just like any mouse can be ordered. There's a minimal cost, of course, associated with it, because now we're in the post-development phase. We now have to pay for keeping the mice alive on the shelf. They are cryopreserved also. So if the mouse, you know, that we initially see a wave of interest in the models as they're newly reported, and then it kind of tapers off as most of the early researchers have already acquired the mice, but they're always recoverable from cryopreserved stock. We cryopreserve sperm and embryos where necessary. And at that point, you know, that can be where the role of a Jackson lab stops. We've made the models, we've reported them. Hans's models would be delivered to Hans, and then it would be up to Hans to decide what he wants to do with them. We're not, unless Hans gives us permission, we're not able to distribute the 2 models that we've made under a specific statement of work, rather than using public funding to make the mouse. And so the lab can then just kind of step away and wish you well, or the foundation or any scientist can come back to us and say, look, you know, there's these levels of analysis that we want to do that our individual research community doesn't have the capabilities to handle. We run what we call a mouse hospital. Essentially, it's a complete evaluation of neurological function through histology. We can look at behavior. We can look at age-related effects serology, as well as look at RNA-seq and transcriptional profiling of different tissues at different times. We can look at embryos and see how they develop and how the embryo is impacted by the mutations. So those capabilities are open to all of you as sort of foundation members in the community, and that would be a discussion with my in vivo pharmacology teams in terms of what is it that you want to do and what's your data set, and what do you want your data set to look like. So we've supported many foundations in that. If the foundation is interested in really looking at the transgenic model again, that could be something that we could also look at one more time and try to develop. We've already developed essentially 2 models with public funding, so it gets a little bit iffy in terms of we want to try to spread the money around as much as we can to different foundations. And the hope is that researchers using the models here can apply for grant funding to either make a model themselves or apply for the money to make a model and then just work with us to make the model for them. Very much how Hans worked with the foundation. The foundation gave you money, and then Hans was looking to outsource that, and we were happy to really be involved in that project as well.
So can I, are there other questions that are burning questions, or can I ask one at this point? So, okay, I have a question, just a couple of things, actually. I have a list of things I'd like to ask, but the C1186Y, this is a premature, is it a stop codon in terms of it
no, it's a missense mutation. It's a tyrosine that replaces a cysteine.
Okay, so you have essentially, it's targeted then for degradation, and it's very similar to what you would expect to happen in some cases.
So we don't know. Again, that's a kind of a follow-on study. Is what is the effect of that mutation on protein expression? Is the missense protein is it stable enough to be seen in an immunoblot, or is it unstable and really is it behaving like a het null mouse? I think it has some biology, because the transmission of our mice that are heterozygous for the C1186Y allele, there doesn't appear to be any bias. We see a 50-50 transmission, whereas with the het null, we see a bias. So that suggests that the C1186Y is partially compensating.
Okay. So what essentially, just to recap here, what you just described is essentially a very broad and in-depth clinical phenotyping of a strain that we would choose, for example. You mentioned various aspects of, the immune system development and so on. So what actually happens with this data when it's generated? This is then does this become then a paper? Is it
okay,
published?
yes, thank you.
So there are multiple levels in which foundations have engaged with us. We can enter into what's called a sponsored research agreement, in which case the foundation kind of declares their interest in what areas they want to be focused on. Well, what is it that they really want? Is it phenotyping? Is it development of additional models? Is it rescue? And the lab would then kind of come up with a quote based on what we think is the most direct path. But the sponsored research agreement does allow us, if we find a barrier, we don't have to go back and renegotiate everything. We can say, okay, we need to solve this barrier, and here's how we're going to solve it. And so we kind of take a little detour. And what happens there is that the material, the data generated, belongs both to the foundation and also the Jackson Lab. And we can support publication by foundation scientists, or we can publish it with the support of the foundation. There are other mechanisms, such as sort of fee for service. Essentially, you can think of, in that scenario, you can think of the Jackson Lab behaving as a contract research organization. We do what you tell us to do, and you own the data.
Okay. Okay, I would have another question on a different topic, if I may, just that you discussed before. The strain that you're developing for Hans, in collaboration with him, this, in principle, the heterozygous knock-in allele should behave in a similar fashion to the there's, for example, the clinical allele or deleted exon 2, right? You would expect it to behave like a head. And
we would expect it.
Did you mention that these mice are also smaller, active and smaller?
The only small mice we see are the C1186Y. So
Okay.
that was a good point. Thank you for bringing that up, Andrew. We don't see smaller mice for the loss of exon 2. That suggests that the C1186Y exerts at least a partial dominant effect. It's a new phenotype, which we don't see as a consequence of the absence of the protein. Now, we haven't done protein level evaluation, so we don't know if there's compensatory effects from other places, but certainly, like in the het null mouse, you can imagine the wild type allele could be upregulated, so the total level of protein is consistent. And that may not happen in a mouse which is heterozygous for the C1186Y. But the small size is unique.
Yep. Could it also be the case that the truncated protein is actively interfering with the function of the remaining wild type protein?
It's probably not truncated. It has a single amino acid difference. It's probable that that single amino acid difference, a cysteine to a tyrosine, is fairly aggressive in terms of effects on localized protein architecture. It could change protein shape in such a way that it's interfering with wild type function.
Right, so it could have essentially some kind of a sort of a semi-dominant negative kind of an effect.
It's perhaps, or you could have dominant and recessive effects going on at the same time. We don't know because the wild type allele is always going to be expressed. Yeah. Now, one way I proposed around that is to cross the mouse to the flux mouse, and then you can get rid of the wild type allele at any time of your choosing, and then that would only leave the misfolded protein around. We do know now in the small number of mice that we've generated that homozygosity for the C1186Y mutation seems to be home lethal. Only 22 mice, I normally like double that, but even with only 22 mice, you'd expect 4 homozygous mice. So the indications are that it's likely to be home lethal.
Okay. I would have another question just in terms of the, the shelf life of a new strain that you generate in JAX. I understand that if there's no interest, from the wider community, you can't keep it indefinitely forever. Is there a time limit that you already know that you keep in mind for when a strain then ultimately moves from, shippable live mice to just cryopreserved and then needing to re-derive the mice?
So that is a business decision which is handled by the folks that are involved on the public side of the lab in terms of selling mice. Typically, what I do know is that if the cost if there's little cost recovery, that is, it's costing us more to keep the mice on the shelves than we can make in the sales of those mice, then that becomes a decision to remove the strains from the shelf to put them into cryo. It is possible that the foundation can support a continued maintenance of mice. They can say, we want to pay to keep these 3 strains on the shelf for 1 year, for 2 years, and in that sense, then we would adhere to the foundation requests, and they would be budgeted accordingly. We do this for complicated models like Rett syndrome and so forth, when there's lots and lots of different clinical alleles. Foundations fund us to make all of the different clinical alleles in the same gene, but they want them all available to the same time, regardless of whether or not they're selling high levels or low levels. And so we can just maintain a cohort of colonies on the shelf, and so long as we get a cost recovery back in terms of the resources required to maintain the mice, we can maintain them indefinitely.
Could I ask if Hans could tell us what he would like to do?
Yes, of course. So our plan so we got these 2 lines now that have the mutation that can then be knocked out with tamoxifen. So we've ordered a tamoxifen Cree mouse, so a mouse where the Cree is only released into the nucleus when you give them tamoxifen. So that allows us to give the mice to rescue the mutation, remove the mutation in postnatal life, and ask whether some of the phenotypes that we see, whether it's growth, neurobehavioral, or others, whether some of them can be rescued kind of as the mice becomes an adult, and if it doesn't work as an adult, we'll move, younger and younger to try to figure out kind of the window of opportunity of, could we treat Wiedemann-Steiner, after birth? That's the big question that I have. So.
I apologize. I do have a hard stop at the hour, but I just want to say I think you've got a good step forward. You've got options and several lines to study, and I think the next step is obviously what Hans is alluding to, is the evaluation. I think there's progress, and that's exciting. I think you've got a good opportunity in front of you to hopefully identify something that can capitulate what will be something of value for drug development, and hopefully translate to something that can be utilized in the clinical group. So that's my comments. I appreciate you letting me join. I'll sit on for another minute, but I apologize. I do have to jump off.
Ciao, Chris. Bye.
I know you mentioned, for example, we haven't done the characterization yet for some of the different mice, and not all of them are going to you, Hans, right? So does it make sense to do characterization of the phenotyping of those mice through that program you said, and from the perspective of the scientific advisory board, or is that something that?
I mean, from my perspective, it makes a lot of sense to do it. I think this is something that we should absolutely discuss, and we can discuss with Aamir what that entails. But for me, I think that this would be. I mean, it's a first step, is it not? I mean, I think there's also probably we need to be careful not to run into too much overlap with what Hans is doing, because I assume he'll also give the mice a good look over and see what he can see what comes out from there. But we should certainly have a look at what JAX can do for us as the next step, and I would support that, definitely.
Can I clarify too? I guess Andrew or Dr. Zuberi, we know the symptoms for Wiedemann-Steiner is so broad and diverse. And we always say in the community, not one kid is like the other because of just the various symptoms. And so I think many of us in the community are curious why that is. Is that something that can be explored with the type of mutation? And I know you.
Certainly, I think you raise a really good point, and we see this a lot. Patient variability. Of course, there's the patients also in slightly different environments, and so we don't know what effect that environment might have on them. But it's almost certain in the background genes play a role, and it's important to realize that all of these mutations are being made on a single inbred mouse strain. So you have to think of it as one patient. And we can generate multiple copies of that, but it's the same patient over and over again. So where we're seeing home lethality, it might be that if we look at when we cross that mouse to different inbred strains, we might be able to rescue some of that lethality. They might live a little bit longer. In some of our models, we've worked out in some of the work which is published, we find that, say, the JAG1 mutation is lethal on the B6 background. But when we change the background, so it's now an F1 for B6 and FEB, which is another strain, that mutation is viable. And so that what that says is that there are some genes on FEB that are rescuing the lethality. Sometimes it's nice to know what those genes might be, because that's another therapeutic option. Maybe you don't have to fix KMT2A. Maybe you need to think about if you can fix a modifier of KMT2A, you might be able to impact the effect of the disease phenotype in a beneficial way also, because we can do it in mice. So there are potential studies that we can outline that address that. Without knowing ahead of time what's successful, it ends up becoming a guessing game. Do you make this strain or that strain or that strain? And it's a little bit of a rabbit hole. You can go down very deep to the point where you're just throwing money and not really seeing positive outcomes. one good way to kind of try to preempt that is try to use our knowledge about how the same gene behaves in different strains to our advantage. And you can look at protein levels, or you can look at mRNA expression levels. And many genes are fairly consistent across strains, and whereas other genes vary across strains. Well, what happens if you cross this strain with a strain which expresses KMT2A at high levels endogenously, or a strain which expresses at low levels? We're still in the middle of exploring that. That was kind of an idea I came up with only a couple of months ago, and we're kind of trying it out. We've got an ALS model that we're trying to investigate and use that kind of approach. If it's successful, I think that we can present it as a possible future for future experiments. The other way is, you know, the source of the variability between individuals could well be that there's multiple mutations in the gene, and exome sequencing will only pick up mutations in exons. There could be intronic effects which are not being recognized, which might have regulatory impacts as well. But I think that represents a real challenge both to the clinical community and also to the sort of model generation communities. How do we mimic that diversity that exists? And, you know, could we think about different experiments? We have models that can do that. It's just right now using mouse breeding to establish that would be very expensive. And at this point, I really wouldn't recommend it as a way to go. I think if we can find out more about the gene and the models that we've made, that might give us a clue as to what these other modifiers might be. There are many other groups that are funded to look at protein-protein interaction, for example. We know that KMT2A is processed in a complex manner. You can think of how the N-terminal and the C-terminal portion of the protein behave, and they're almost, you could consider them as two different proteins expressed from the same gene. So maybe some of that explanation of the phenotypic variation might be due to that, or it might just be due to you know, other effects on background genes that we don't have a good handle on right now.
Yeah, that's really helpful, and I thank you for explaining that. And I know we at the foundation we know we need to do a better job around gathering data with our own community and pursuing natural history studies. And I think that will in parallel help with the work that you're describing too. So
it's true. We were funded by the sort of limb-girdle muscular dystrophy association to make the same mutation in 4 different strains. To kind of explore that effect. All 4 strains behave exactly the same. It tells us how stable the effect of the mutation is, but in that case, it was a deletion. My personal view as a long-term mammalian geneticist is that deletions, big deletions, tend to be very hard to modify, right? If you're missing a biological activity, it's very hard to think of how you might compensate for that absence. Point mutations are perhaps more beneficial when it comes to looking for modifiers. So the C1186Y example might be an example of that. If the mutant protein is stable, and that's a big if, because in some cases, a single amino acid change has resulted in the complete absence of the protein. So it behaves like a null, even though it's just a missense. But if the C1186Y mutation is a stable protein, but misfolded, it might be possible to think of how it might be modified either by another mutation in the same gene or by a partner molecule that might twist that shape back closer to what it should be.
I just wanted to make one comment. You know, now that these strains are available, it is also quite possible that investigators will jump on them and do some of the phenotyping for free. It's hard to know. It's hard to predict. You know, these things kind of come in waves. Certain genes are quite popular at one time, but you know, one thing the foundation could do is see how much interest there is in the strains, and then decide on if there's no interest to contract out JAKs or do other things like that. But it could be that someone will do that for you now that the reagent, it's so, you know, I really believe that reagents dominate kind of how the science moves. And I think we are we are poised to make great discoveries here. I think both temporally, tissue specific, and missense versus other. So I think it's an exciting time, and you know, we currently only have funding for that one question, you know, that temporal, but I think it's a very, very important question. But there's many other cool things that can be done as follow-up. So either at JAX, at my lab, or maybe some other lab. So.
Yeah, thank you for that answer. I did neglect to mention one possible way forward as well. So many investigators have interests in mutations and phenotypes, but they lack resources at their facility. So it's also possible, and we've done this in the past, where they write a grant, and if it and it includes a animal phenotyping component, and JAX does that for them. So it would be essentially we get to do the phenotyping based off a funded piece of work that's been acquired by an investigator who may not have expertise in working with mice, may not have the range of equipment. And so that in that case, that's a collaborative research partnership that we would work under. And even though Hans's initial request, it was as essentially as a fee for service approach, I kind of changed it more into a sponsored research agreement because it added to the work that we were already doing, and I can't let something interesting go by without jumping in with an opinion, and I don't charge for my opinion. So in that sense, it's a sponsored agreement. I think it's a very productive agreement. I'm hopeful that as Hans continues to get funding, we can continue to support his work. But I think that if you can find other scientists who have specific interests, I already documented that the IMPC database shows an eye defect. I don't know if you're seeing that at the clinical level, at a high level, but gene therapy in the eye is already being approached, and so that is likely to perhaps be a successful endeavor. So a researcher with interest in retinal pathology, for example, could jump on this model and use it to write a grant looking at using the mice to study that. So it's important as a foundation. My advice would be, don't only think about people who are only interested in WSS, but are interested in the individual components which are present in the model, and then try to engage with investigators that have a good publication record in that particular arena. Maybe they won't cure WSS, but certainly they might be able to alleviate some of the clinically relevant symptoms that you're seeing in the children and patients.
Yes, I think certainly one step could be that the people who we Hans's competition at the time, people who we've actually, alerted. I remember we contacted a range of different work groups. We should absolutely make them aware, that this is now available and that they can, they should
That's
jump into the first past the post.
Yes, so please do broadcast the strain. I don't have a marketing budget at all. Get out there and tell your families and tell the research communities that you've had any kind of associations with that. Hey, these mice are coming. You can get them from JAX, and that this is an area where overlaps with your known interest, and maybe they'll take it on. The lab, or let me say that the mouse community has tried to make lots and lots of mutants in the past. And I think global distribution failed because there really wasn't a lot of biology attached to them. The big advantages for the models that we make now are that they're connected to a known rare disease, and there's always funding available to study disease-relevant genes. And we try to add enough, confirmation of the biology to the public website, so that they're not just getting a mouse that may or may not be relevant, but they're actually getting a model which shows a phenotype that is consistent with what they want to study.
With respect to those going public, you mentioned even maybe alerting them that they're coming. Is there something that you'd recommend or something you could share with us that we could distribute to the community that gives them that information? Maybe it's even one of the slides that you had in there or something else?
Certainly. I mean, feel free to distribute any of the slides that we have. Some of the work is still preliminary, but I guess if you disperse it with that proviso in mind, the numbers may change as we generate additional information. But right now, if you think that our summary slide reflects the range, you could probably, might recommend deleting Hans's models from that, so that we're just talking about the strains that we're going to go public with. Then no, I have no objection at all. And actually, if it would help, I would encourage it. We can work together on a press release for when they actually do go public. That's another way in which we've worked with that. We can advertise it on our JAC's homepage, and as a foundation, you can distribute it to all of your interested collaborators.
I think that makes great sense. I mean, maybe we can preemptively, like you said, Andrew, share it with those that we know are already interested in some of the preliminary work, and then we could do the press release and then hopefully do some social media charges and other things to help get it out to folks that we think may be interested. So great. Well, I really once again appreciate everything. I know we're already over time that we budgeted, so thanks for sticking around. If there's any last question maybe, and then
I just want to say, I think you've given us, I've heard some say a small step forward. I say this is a huge leap forward for where we were. So thank you. You've given us a lot to think about, and we're going to be excited to discuss it and follow up, I know.
Well, thank you for that. We've certainly, I think all of us that work on, and my team especially, take a lot of pride and pleasure in making these clinically relevant mouse models, and we hope that they prove as useful to you as we hope that they are when we started this out. So I think it's true, but it's still, I don't know, is it the end of the beginning or the beginning of a longer process? To make the mouse models, we've got to a point now where it's become more routine. It's not as big a challenge as it was even 3 or 4 years ago. Every year brings new improvements. Having established the models, the real push now is, how can you fix the mutations? How can you look at the phenotypes and rescue them? That's, I think, is going to be the clinically relevant progress. And hopefully you can bring in enough clinician researchers and other teams. We do have contacts with folks that are interested in AAV9 may not be appropriate just because of the size of this protein if you're looking to rescue it, but maybe lentivirus could be an approach. We have some contacts with research teams that we collaborate with. I've also got a $500,000 grant that was funded for this year to look at base editing. It's a different way of utilizing CRISPR to make specific rescue mutations in the mouse. There's a lot unknown about that technology. Primarily, the major concern is the specificity of the change. We want it to go to a particular region, a particular gene. We don't want it randomly changing other regions of the genome. In that case, the cure would be worse than the disease. And so hopefully with NIH funds, and I've got 1 year to spend $500,000, so it's going to be hectic for a while. But if we can make any progress in understanding how to improve the vectors, how to improve delivery, you don't need to have the whole gene then. You just need to have enough of it which guides the base editor to that region of the protein to change that mutation. Not all mutations will be amenable to base editing, but where possible, that might be a very good potential future therapeutic. But it's all still in the experimental stage, but we're trying to think ahead. We're trying to get to a world where no disease is left untreated.
I have a recommendation for you. You can use that $500,000 to try to correct the C1186Y mutation and see if you can.
Unfortunately, the grant. If our work is successful, I think that there's lots and lots of possibilities to write smaller grants more that drive this.
A potential starting point for you guys.
Yeah.
Well, very much appreciate everyone's time. And Dr. Zuberi, as you're thinking through, these opportunities, please, feel free to reach out to myself and Christina, who are leading the scientific kind of advisory board work, but also sit on the foundation board, so we can make sure that as opportunities present themselves, we can reach out and bring them to the scientific advisory board and bring them to our board of directors and hopefully make some decisions that are going to be in the best interest of the whole WSS community. So thanks again for everything. I mean, you all are doing so many great things. So appreciate it greatly.
Thank you all for your time. Bye-bye.
Thank you so much.
Bye. Thank you.
Bye-bye.